Enzymes are specific types of proteins that are used to speed up chemical reactions. In the diagram to the right, the parts of that reaction are described. The substrate is the substance that is being acted upon to either break down or put together. The enzyme, shown in light blue, is there to fit together with the substrate to form an enzyme-substrate complex. Fitting together causes that substrate to weaken its bonds and break into products. Each enzyme has one particular shape of substrate that it will fit with. The shape of the enzyme determines its function. The picture to the bottom right shows that enzymes come in a variety of shapes, each one specific to the substrate that they are acting upon. So only one enzyme will work with one type of substrate. In the liver power lab, the substrate being acted upon was hydrogen peroxide. The enzyme was catalase. When the hydrogen peroxide and catalase came together, the peroxide broke into oxygen gas and water as products. The enzyme was not changed in the process, and catalase is free to go and catalyze or speed up more reactions with more hydrogen peroxide molecules. Enzymes speed up reactions by lowering activation energy. The activation energy is the energy needed to start a reaction. The reactants listed on the left are similar to the hydrogen peroxide or burning food that we did in class. The reactants to proceed to products have to have a certain amount of energy input in order for the reaction to proceed. When an enzyme is added, the dotted line, that amount of energy needed for the reaction to proceed is much lower. So enzymes lower the activation energy or reduce the amount of energy needed for the reaction to begin, and thus the reaction happens faster. Several things can alter the shape of proteins or enzymes, and these denatured proteins then have a difficult time finding or matching with the substrate to continue the reaction. Heat can denature proteins, and so can pH. When the catalase enzyme from the liver was placed in an acid pH, the enzyme changed shape and was unable to catalyze the reaction of hydrogen peroxide breaking down. In this example, the graphs show the optimum or ideal pH and temperature for the catalase enzyme. The optimum pH is approximately 8, shown by the high point on the graph to the left. The optimum temperature in which this enzyme works is approximately 40 degrees Celsius. Both of these graphs of activity will be impacted as the pH or the temperature change. For example, in the optimum temperature, if the temperature decreases to 20 degrees Celsius, the enzyme catalase will change shape and not be able to react with the hydrogen peroxide. The example looked at in lab involved a change in pH. When the enzyme catalase was placed in an acid environment, the enzyme changed shape and was unable to catalyze the reaction with hydrogen peroxide. Enzymes are important for all reactions in the human body, and the shape of the enzyme determines its function.